So all the accolades go to me, all the hard questions, point them to Mark, please. So thank you for the invitation. For those of you who are not familiar with Juventas, we're a privately funded venture-backed company in Cleveland, Ohio. We started in 2007 with an exclusive license from the Cleveland Clinic to a portfolio of IP that Mark had developed while there. Foundation of the company is really around molecular factors that activate or enhance natural stem cell-based repair. And what I'm looking for today is showing you some data that we've now generated from a couple of phase 2 clinical trials. The story really starts about 15 years ago in Mark's lab with a hypothesis that he had, which is stem cell-based repair is a natural process that exists in all mammals, but it is clinically inefficient not because we lack stem cells, but because the molecular drivers that regulate that system are dysregulated. And he set out with the hypothesis that if we could identify those factors, they themselves could become drugs rather than the stem cells themselves. 15 years later, what we now know based on research from his lab as well as dozens around the world is that SDF1 is the key regulator for activating stem cell-based repair in a range of end organ systems. So Mark's work obviously started in the heart as a cardiologist, and that's where we're focused, but the biology of SDF1 and its receptor CXCR4 have now been replicated in the liver, the kidney, the brain, the skin, the vasculature. It's really a very well-conserved axis that's able to promote repair through a number of mechanisms. So we know that it recruits bone marrow-derived cells to sites of injury, tissue-specific stem cells, promotes angiogenesis, and reduces scar formation. And it's really bringing together these mechanisms that help promote significant end organ repair when you introduce SDF1 at a time remote from injury. At Juventas, we're focused on delivering SDF1 through a non-viral DNA plasmid strategy. JVS100 expresses for about 3 weeks and is when delivered to the targeted tissue. We like the non-viral DNA strategy because what we know is it's safe, it's highly scalable, it's stable for long periods of time, and the COGS are nominal. We're talking about a handful of dollars for our highest doses. We've been showing this slide for years, and it really, I think, best summarizes what we're trying to do as a company. Our goal is to take what is a process of cell therapy and transition it into a vial. And I think hopefully we'll show with our data and where the field is moving, there is a general appreciation that what these cells are doing is delivering factors, and we believe SDF1 at JVS100 really is a key driver in that stem cell-based repair pathway. Along the lines of that, the field is also starting to demonstrate the importance of SDF1 in promoting repair. So this is from a publication that was put out in JACC, I think a couple of months ago, based on an analysis of cells that were used in the Caduceus trial. So for those of you who are not familiar, Caduceus was a phase 1B study where they used autologous cardiospheres to treat patients about 1 year post-MI, or within the year after an MI. And what they were able to show was the delivery of the autologous cells led to a significant reduction in scar mass as measured by MRI. Eduardo Marban, who's the founder of Capricor as well as a founder for the company, went back and he did gene arrays or analysis to look at what genes were expressed by those cells to see if there was any correlation between different products and the reduction in scar mass. And what was demonstrated was that the only protein that correlated with benefit was SDF1. This is consistent with a lot of work from Mark's labs and others showing that a major effector of cell therapy is through SDF1, showing that it not only has recruitment effects but also has direct trophic effects. At Juventas, we've primarily focused on heart failure, and so this is just showing that we're a very active company. You can see that we have 2 heart failure trials. What I'll spend most of my time today talking about is STOP-HF, which is our fully enrolled heart failure study. We also have a fully enrolled critical limb ischemia study. I'll share some data with you from that as well. I think the important takeaway from here is that we've now treated over 150 patients with JVS100, and we have absolutely no significant side effects. So I think we're demonstrating clean safety, and what you'll see hopefully is that we're having nice signs of biologic activity as well. So to focus on heart failure, which is where we spend most of our time, we've treated over 100 patients in heart failure so far. We've completed a phase 1 open label. I will just quickly go over that data with you. As mentioned, we have the STOP-HF data, which is demonstrating some nice signs of structural improvement within advanced heart failure patients. And we have another ongoing trial called RETRO-HF, which is a similar heart failure population, but what we're doing is validating an alternative delivery system to create some flexibility for ourselves. So our phase 1, which was published, I think, in maybe last year in Circulation Research, was a 17-person open-label study. We looked at 5, 15, or 30 milligrams of JVS-100 delivered in one procedure to patients with NYHA Class 3 heart failure. So these are patients with significantly advanced heart failure, fatigue through normal daily activity. All the patients in the trial were on optimal medical therapy, had an ICD, and I think at least 30 to 40% of them had BiV pacing. What we saw in that population was we met our safety endpoint. 18 months later, 15 of the 17 patients were still alive. What we saw from an efficacy perspective, respecting that it was an open-label phase 1, were dose-dependent improvements at a year in BNP, quality of life, and 6-minute walk. What that did was allow us to start a phase 2 trial last year called the STOP-HF trial. PIs of this were Warren Sherman from Columbia, Les Miller from Pep and Hart, and Jean Chung from Lindner. Again, we were looking at a ischemic symptomatic heart failure population. It was a placebo-controlled randomized double-blinded trial design. We looked at controls, so patients actually received 15 injections of placebo, 15 milligrams of drug, or 30 milligrams of drug. And the drug was delivered using a Biocardia helix catheter. So this is an endoventricular injection catheter that delivers in about a 45-minute procedure the drug around the infarct area. Here you can take a quick look at the type of patients we were treating. Mid-60s, I think of note, they were on average around 11 years removed from their heart attack. So this was a pretty advanced heart failure population. If you look at their clinical parameters, more than half of them were NYHA Class 3, 6-minute walks of less than 300 meters, poor quality of lives. You can see based on ESV, their hearts were significantly enlarged, about 3 to 4 times normal. EFs were significantly depressed, under 30, and they had elevated NT-proBNPs, which is a marker for cardiac stress or cardiac remodeling. So, and we enrolled this at 16 centers in the US. Again, it was delivered and this will get back to the other delivery system we're also evaluating, by the Biocardia Helical Infusion Catheter. It's actually CE marked in Europe. It has been used in over 235 cases. It's worth noting that we effectively delivered drug to all patients, and we had no unanticipated serious adverse events. So it really is about a 45-minute procedure that can be done in a wide range of institutions by an interventional cardiologist. Here's just a summary of the data from all the patients. What you'll note, and it's in our high dose relative to placebo at 4 months, p-values on the right-hand side. What you'll notice, the preponderance of the data is showing a benefit of 30 mg relative to placebo. What we saw were clinically meaningful benefits in all of the clinical parameters: 6-minute walk, quality of life, exercise time. Not unexpected, at a 4-month time point, we did see a significant placebo effect as well. So some of these placebo patients improved, just not to the magnitude of the treated. I think comforting to us is that on the objective parameters, such as ESV, EDV, proBNP, we saw the most significant and consistent changes. It's always been a belief, well, I should say it's always been a belief of Mark's in the field, that these regenerative therapies are most effective in patients that are the most at risk. So we know this really from, at Juventas, our pig studies, in which we were able to demonstrate that JVS-100 had the greatest impact in pigs with the largest infarcts, but also from the AMI field, which in the early days, as they started evaluating cell therapy in AMI, observed that the greatest benefits were seen in those patients that had STEMI MIs with EFs under 40. Fortunately, this trial was large enough that we could actually stratify the population into those that were moderate risk and high risk. And we did so using end systolic volume, which is appreciated as probably the best predictor of outcomes in this patient population. So here you'll see there's about, I think about 11 to 14 patients per cohort here. You'll see on the clinical parameters that they're relatively consistent, but the real changes occur when you look at the volumes. So we're looking at 117 mils in the moderate risk patients, about 225 mils in the high risk, significantly increased EDVs, and the EFs go from low 30s to almost mid 20s. So this high-risk population really is what we call the frequent flyers. They're driving the most significant healthcare costs. When we look at our patients that had the greatest number of hospitalizations within a year of treatment, it falls within this population. What we see when we look at the high-risk patients now is, first of all, two things comfort me about this data. One is the consistency in all the objective parameters, and the second is that we see a clear dose-dependent curve in every single parameter we look at. We're seeing almost a 24 mil improvement in end diastolic volumes, and we're seeing almost a 31 mil improvement that now reaches significance in end systolic volumes. And when we look at that as a function of change in ESV against baseline ESV, we really do see a very nice dose-dependent trend towards patients with larger hearts having more significant pronounced responses to JVS 100. I think this gives us a couple of important pieces of information. It shows us, first, that we have an active drug. I think the second thing, it really shows us where we can have the largest clinical impact. And fortunately for us, it's in a patient population that has the greatest need. Now, it's worth noting that I don't want to suggest that the lower or the moderate risk ESV patients will not benefit. What we saw in our phase 1, at least what was suggested, is we'll preserve function in these patients. And while we won't see the significant reduction in ESV, what we'll see is a maintenance of function over time. And we're excited by this because if you really do look at the literature and what drives outcomes and is the best predictor of hospitalization and mortality in these patients, it is ESV. With the 30 mil changes that we're looking at, meta-analysis would suggest that we will have somewhere in the ballpark of an 80% probability of a positive mortality trial as we look to future endpoints. The other thing that's comforting is all of the objective parameters showed these benefits. So EF, provided is a function of volume, also showed a benefit, as did changes in BMP. As you'll notice, the placebo group worsened by about 750 picograms per mil. Clinically relevant change is around 250. So these patients really are in a dynamic state and worsening, and what we appear to be doing is, in the treatment groups, improving volumes and stabilizing the patients structurally. So getting back to that original slide where we talk about turning process into product, I think we're getting closer. We're demonstrating that now in a clinically meaningful way. What we've done here, we've lined up the data from our STOP-HF trial against some recently published data. So MSC heart failure was an autologous MSC trial that was presented as a late breaker at ACC. The C-CURE trial is the cardio-free trial using autologous cells as well. And the Caduceus trial I mentioned was a post-MI trial. And I think clinically we're now able to comfortably state that we can induce benefits equivalent to cell therapy by delivering SDF1, and that SDF1 can activate these endogenous pathways. So just to conclude on the heart failure data, and I see Chris looking at his watch, so I better hurry up. We believe we have an active drug. We're having benefit on all the objective parameters, and we're doing so in a patient population with the greatest need. We do have another heart failure study looking at an alternative delivery system. This comes up through the coronary sinus. It shortens the procedure about 15 to 20 minutes, and it's using off-the-shelf balloons and catheters. What's nice about that is it can now be performed not only by interventionalists, but by EPs and heart failure specialists as well. Again, it's a 72-person phase 1/2. We've now enrolled about 45 to 50 patients in that trial. I can tell you the procedure is running very smoothly. We've safely delivered drug. We've really had no complications there as well. One of the exciting things for, I think, all of us in the field is that the biology of regenerative medicine really does translate from one organ to another. So I just want to quickly, and using mechanisms that I think we all appreciate and have discussed, I want to share with you some preliminary data that we have, or interim data, from a CLI study. So this was a first-in-man phase 2A study where we looked at, in a double-blinded, placebo-controlled dose escalation study, treatment of JVS 100 in Rutherford class 4 or 5 patients. So these are patients with late-stage PVD, significant ischemic pain or open ulcers, and high risk for amputation. Again, patient population is relatively standard for this group. We enrolled half the trial in the US and half in India. What we saw was, and this is interim data, so we're still blinded, but we saw a dose-dependent improvement across all quality of life parameters And this was not powered for any kind of significance, but we're seeing some nice trends there as well. This correlated with significant reduction in VAS pain scores in, again, a dose-dependent fashion, as well as significant improvements in wound closure. Mind you, in the baseline, there's an imbalancing of the wound size. That's just due to randomization, but we do see those wounds close significantly. We'll have full data from this coming out sometime in the third or fourth quarter of this year. What's interesting for us is that, at least in this trial and what we're hearing from others, this amputation rate has gone down significantly, in part by a more aggressive approach by vascular surgeons and more awareness of the disease. So we actually have had an incredibly low amputation rate, but I think from a product perspective, the fact that we're again seeing a clear biologic sign and that we're having consistent benefits in some clinically meaningful parameters suggests that there's a path forward as this potentially being an adjunct to below-the-knee stenting or procedures that vascular surgeons are already doing. We also have, and in the interest of time, I won't share with you, but we have data showing that JVS-100 is able to reduce scar formation post-surgery. We've been developing that through a spin-off company called Seronarex. Data is quite nice. We also have preclinical data showing muscle regeneration as well as improved incontinence. So I think we're very happy to be able to demonstrate that we can use a factor-based approach to elicit responses similar to cell therapy. We've done so clinically in a couple of indications. Preclinically, we've got proof of concept across multiple indications. What we're looking forward to now is 12-month data from our heart failure study at the end of this year, full 1-year data from CLI around the end of this year. And in total, I think around the beginning of next year, we'll have over 200 patients worth of clinical data. So hopefully next time I'm talking to you, I'll be telling you a little bit more about where we are. Thanks.
Thank you.